In the winter of 1943, an embargo and a harsh climate created a severe famine across the Netherlands. It became known as the Dutch Hunderwinter, a historical tragedy that unintentionally provided researchers with a stark, natural epidemiological experiment. For 8 months, daily rations plummeted to roughly 1,100 calories and just 34 grams of protein. Pregnant women endured these severe biological stressors during critical periods of gestation. The immediate clinical outcomes were exactly what doctors expected. The average birth weight dropped by over 13 ounces, and the delivery rate of low birth weight infants spiked by 50%. Decades later, researchers tracked down the adults born to this specific cohort to measure how that early deprivation affected them. The data revealed a startling pattern of long-term health consequences. This chart maps birth weight against later risk of cardiovascular disease. The baseline risk remains flat for heavier infants, but as birth weight drops below the 5-pound mark, the relative risk of adult heart disease jumps by 50%, and stroke risk more than doubles. These findings formalized a framework known as the Developmental Origins of Health and Disease. This framework proposes that environmental exposures in the womb permanently shape a person's lifelong risk for illness. Chronic conditions like hypertension and type 2 diabetes do not spontaneously generate in adulthood. They are actively programmed in utero in response to the environment the fetus encounters. For decades, popular wisdom assumed the fetus acted as a parasite, extracting whatever nutrients it needed directly from the mother's body, even if the mother's diet was completely inadequate. Biological reality enforces a strict triage hierarchy instead. Available nutrients are used first to ensure maternal survival, second to maintain placental function, and only third to support fetal growth. Maternal and fetal bloodstreams do not mix. The chorionic villi act as a strict barrier, consuming up to 40% of maternal glucose. When calories drop, the placenta restricts nutrient transfer, fulfilling its own needs first to prevent the mother's reserves from fatal depletion. Fetal malnutrition is a deliberate, highly regulated survival mechanism executed by the placenta to protect the maternal host from starvation. Under normal, well-nourished conditions, fetal growth follows a genetically predetermined schedule, operating at a faster rate than at any other point in human life. This growth occurs in 2 distinct phases. The first phase is hyperplasia, which is the multiplication of entirely new cells, and the second is hypertrophy, which is the physical enlargement of those existing cells. Hyperplasia does not happen continuously. Organs and tissues multiply their cell counts only during strict, pre-programmed time windows, known as critical periods. Here we see the normal hyperplastic trajectory of the human forebrain. Cell multiplication is measured by tracking total DNA content, which spikes rapidly in this specific window between 10 and 20 weeks of gestation, and then naturally levels off. When severely undernourished, this process is stunted. A healthy baseline curve rises sharply, while the undernourished curve remains depressed, indicating a severe cell count deficit. Because this biological timer expires, postnatal feeding cannot trigger new hyperplasia. The total cell count never reaches baseline. Critical periods are a biological one-way street. Missing the narrow window for hyperplasia guarantees a permanent, lifelong structural deficit in the affected organ. In clinical settings, these deficits result in infants classified as small for gestational age, but the timing of the starvation dictates the exact nature of the damage. Infants who suffer early chronic malnutrition are born proportionately small, with a permanent reduction in total cell count across their organs. Conversely, those starved during the 3rd trimester are born disproportionately small. Their organs have the correct cell count, but they are completely depleted of late-stage glycogen and fat stores. To survive this severe intrauterine starvation, the fetus deploys an ultimate metabolic defense strategy, a process researchers call the Thrifty phenotype. The fetal triage system prioritizes the central nervous system. To keep the brain alive on restricted calories, the fetus sacrifices its own skeletal muscle tissue. The fetus achieves this through epigenetic modification. By adding specific chemical markers, the fetus alters how its genes function and express themselves without altering the underlying DNA sequence. To divert energy to the brain, the fetus epigenetically silences genes producing insulin receptors on its muscle cells. The muscle tissue becomes intentionally insulin resistant. It refuses to absorb glucose, forcing the vital fuel to bypass the muscle and reach the developing brain. This induced insulin resistance successfully keeps the fetal brain alive until delivery. As a result, the infant is born with a metabolism permanently programmed to hoard calories and resist insulin. That metabolic programming persists into adulthood. An individual who survived an in utero famine via the Thrifty phenotype reaches maturity with a biology wired entirely for starvation. The crisis begins when that carefully calibrated scarcity metabolism is placed into a modern environment of continuous caloric abundance. Biologists call this the mismatch. When this adult consumes a high-calorie diet, their bloodstream is flooded with glucose, but their muscle tissue, still operating under epigenetic instructions, refuses to absorb it. The pancreas overproduces insulin to force the glucose into cells, creating a cascading systemic failure. This direct biological collision results in the rapid onset of adult type 2 diabetes and hyperinsulinemia. Similar epigenetic changes made to cholesterol metabolism during the famine also remain active. When exposed to modern high-fat diets, the adult body fails to produce sufficient HDL cholesterol, triggering severe atherosclerosis. Human gestation is the critical programming phase for our lifelong metabolic software. The exact nutritional environment a fetus encounters shapes its biological responses for decades to come, making maternal nutrition the ultimate defining form of preventative medicine.